Students, have you ever thought about the fact that we are constantly exposed to the many billions of the viruses and bacteria per second? Have you ever thought that why we are not infected by those viruses and bacteria? That's because of the reason that we are having an army of thousands of cells patrolling in our body that is defending us from these viruses and bacteria. So in this lecture, we are going to discuss in detail of these cells and tissues that are our army of our bodies. So let's discuss it. So what are the learning objectives for today's lecture? These are the different types of cells involved in the immune response that we call the army of the cells. What is the major histocompatibility complex molecules, MHC? What are the antigen presenting cells? What are lymphoid organs and cytokines? So cells, aka army of our body. These are of three types. These are lymphocytes, antigen presenting cells, and various effector cells. We will discuss each and every cell in detail. First of all, lymphocytes. These are present in the circulation in various lymphoid organs. These are of two types, B cells and T cells. T cells mature in thymus while B cells mature in bone marrow. T suffix shows before lymphocyte that these are mature in the thymus, while B shows that these are mature in bone marrow. These cells are generally programmed specifically for antigen that is done by joining and diversification of different gene segments to form functional antigen receptor genes. So they are having the specific receptor for complementary antigens, mean it binds only to specific type of antigen. This is done by their diversification and joining of different gene segments that forms the functional antigen that is specific for only one type of antigen. So therefore they are lymphocyte fall in the category of specific type of immunity. We have also discussed specific and non-specific type of immunity. The receptors that are possessed in B cells are called antibodies. B cells, they are having the receptor on their surface. We call these receptors antibodies, while receptor on T cell called T cell receptors. Mature T cell and B cell recirculate through secondary lymphoid organs and they bind to and activate the clones of lymphocyte that express receptor for those antigen in a process known as clonal selection. So these cells, T cells and B cells, when they are formed in the thymus and bone marrow, they are not specific to any type of antigen. They are really programmed in the secondary lymphoid organs where they are exposed to the specific type of antigen. When these are exposed to these antigens, to specific antigen, T cell receptors and B cell receptor forms clones. They multiple divides. Mitosis occurs, causes clones of B cells and T cell form that are specific to that antigen. We call this type of process as clonal selection. What are T lymphocytes? These thymus-derived T lymphocytes mature in the thymus after developing into bone marrow and develop into effector cells of cellular immunity that help B cells to produce antibodies. The help is highlighted here. We would discuss this help in the types of T cells, such as there are four types of T cells and their functions are T helper. This secret soluble molecules, we call it cytokines, that stimulate both B cell and macrophages. B cell, when stimulated by T helper cell, releases antibodies, while macrophages performs their phagocytic action. These are stimulated by T helper cell. Therefore, it is called helper cells. While cytotoxic T cells are the cells that destroy infected cells by directly binding to that cell, therefore called cytotoxic, means injection of the toxic substance into the infected cells. While T regulatory or suppressor cell has an important function in suppressing excessive immune response. So there must be cells that should limit the immune response, so that our self tissues should not be destroyed by excessive immune system. So therefore, there are the regulatory cells that have important function in suppressing immune response when there is no need for immune response. The natural killer cells, these are the cells of innate immunity. These recognize microbial products regardless of MHC. We will discuss MHC molecules in further lecture of MHC and may play important role in defense against infections. So there these in this chart, in this picture, you can see the function of every lymphocyte. There are the B lymphocyte that when bind to the microbe, then when exposed to antigen or any microbe, they bind to it, they release antibodies from the surface, such as in this diagram shown. When antibodies are released, they either act by direct action or by initiating complement system. We will discuss complement system when we discuss B cell or humoral immunity in detail. So antibodies causes neutralization of microbe, phagocytosis or complement activation. Now here are the T lymphocytes. There are 4 types of T lymphocyte and the function are shown. Helper T cells, these microbes, these recognize the antigen presented by antigen presenting cells. Antigen presenting cells we will discuss in detail. So when T helper cell recognize the antigen presented by antigen presenting cell, they would release cytokines that cause inflammation. They also release cytokines that activate macrophages and causes activation and proliferation of T and B lymphocytes. So T helper is the main cell that causes the immune response to occur. It causes activation of macrophages, inflammation, and activation of T and B cells. So all the humoral immunity and the T cell mediated immunity is dependent upon action of T helper cell. Cytotoxic T cell, they recognize the antigen presented by microbial antigen by any infected cell. They bind to the infected cell. They destroy by releasing many proteolytic enzymes, therefore called cytotoxic T cell. Regulatory T lymphocyte, they suppress the clonal expansion and action of B and T cells. Natural killer cells also directly kills the cell. So T helper and T cytotoxic cells. T helper is also called CD4 plus. Why they are called CD4 plus cells? Because they express T cell receptor of CD4 plus class. On T helper cell, there are the receptor present that sense the presence of antigen, such as these receptors are there. These sense the antigen presented by antigen presenting cell. What these receptors are called? These are called CD4 plus. So therefore, T helpers are also called CD4 plus cells. While cytotoxic T cells also called CD8 plus because they express CD8 plus T cell receptor on the surface. T helper has CD4 plus receptor. Cytotoxic T cell has CD8 plus receptor, while B cell has antibody expressed on their surface. Major histocompatibility molecule MHC. The function of MHC is to display peptides for recognition by CD4 and CD8 plus cells. Its function is to just serve the food to CD4 plus and CD8 plus immune cells such as helper and cytotoxic T cells. It basically displays antigen that are the foreign substances to these cells. T cells sense only the peptide fragments of the protein displayed by molecules of major histocompatibility molecule. So all the T4 types of the T cell only they are activated when they find or when they bind to the antigen molecule presented by MHC protein. So the human MHC is called human leukocyte antigen complex HLA. There are two types of MHC, MHC class 1, MHC class 2. What is the function of MHC class 1? These are expressed on all nucleated cells of the body. When there are, these MHC class 1 proteins are present on all nucleated cells of the body. They display antigen only to CD8 plus cells. When MHC class 1 express antigen, mean it binds antigen to its surface, it only activates CD8 plus cells that are cytotoxic T cells. While MHC2 cells are only expressed to antigen presenting cells. These are dendritic cells, macrophages, and B cells that display antigen to only CD4 plus cells that are helper cells. So MHC class 1 activates CD8 plus cytotoxic T cells, while MHC class 2 activates CD4 plus cells that are helper cells. So here is the expression of CD4 and CD8, sorry, expression of class 2 molecules and class 1 molecule, MHC class 1 and class 2 molecules. Here they are bind to the plasma membrane. They express peptide fragment of antigen, and there is a binding site for the CD. So this is class 2. So there is a CD4 plus binding site here, while MHC class 1 molecule has also the peptide fragment bind to it and also the peptide binding cleft. That is the activation site, that is a binding site for the class CD8 plus molecules. So B cells, these are the bone marrow derived cells or lymphocytes that produce antibodies that are mediators of humoral immunity. B cells are involved in humoral immunity. These are also present in lymphoid follicles of peripheral lymphoid organ that are called secondary lymphoid organs. Antibody of IgM class is expressed on cell surface that form B cell receptor complex, as we have discussed previously, that B cell has a receptor that is formed from the antibodies which are of which class? IgM class. They do not require MHC molecule to be activated, so they do not require, they directly bind to the antigen and causes release of antibodies. When activated, it differentiates into plasma cells that secrete antibodies. Types of antibodies secreted, these are 5 types of antibodies: IgM, IgG, IgD, IgA, IgE. IgM is a major function in elimination of foreign antigens, and it has also function in primary response. Primary response is a response in which antigen is first exposed, first time exposed to the immune system. IgG antibodies have role in secondary response when same antigen is exposed second time, then the immune response occurs. So IgD antibodies form B cell receptor complex. Also IgM class and IgD form B cell receptor complex. IgA released with mucosal secretions, and IgE has a role in allergic reactions. So natural killer cells, these are the innate immune cells, as they are functional without prior activation and do not express highly variable and clonally distributed receptors for antigen. So they are not specific to any type of antigen. They, when activated, they kill all the cells that they are exposed to. They are like the hitmen. So when they are activated, they kill all the cells in when regardless of their specificity. So two types of receptors are there, inhibitory and activating. The inhibitory receptor recognize self class 1 MHC molecules. So class 1 MHC molecules are expressed in all nucleated cell as we have discussed. So inhibitory receptor recognize class 1 molecules and differentiate self from non-self antigens, which are all, as we have discussed, exposed in all healthy cells. Whereas activating receptor recognize molecules that are exposed or unregulated on stressed or infected cells or with cells with DNA damage. So when there are cells with DNA damage, such as mutation in DNA, such as in cancer or infected cell, virally infected cell, inhibit activating factor, activating receptor on natural killer cell recognize these type of molecules expressed on these infected cells and kill them. So antigen presenting cells, as the name describes antigen, they present antigen to the CD4 plus and CD8 plus. The immune system contains several cell types that are specialized to capture microbial antigen and display these to lymphocytes, such as T and B cells. These cells consist of dendritic cells, macrophages, and B cells. Dendritic cells are the most important antigen presenting cells for the initiating T cell response against protein antigens. So what is the function of antigen presenting cell? Basically, they ingest, they phagocytose, such as macrophages and dendritic cells phagocytose the incoming microbes. They process it and they display the fragment of their antigen on class 1 and class 2 MHC molecules. This is the function of APC, such as in this dendritic cell with antigen. This dendritic cell is exposed to antigen and it really expresses antigen on its surface. It comes with the lymph flow inside the lymph node. There are T cells and there are T helper cells and T cytotoxic T cells. They are never exposed to any antigen. They are called naive cells. NAIVE, naive cells. They are never exposed to antigens. So dendritic cells work as a antigen presenting cell. It presents antigen to both of these cells. When antigen is presented to these cells, clonal selection occurs, formation of clone of these two cells. They now circulate into they are now called differentiated effector cells. Memory cells are also produced in this process, such as described here. These are the naive T cells when they exposed to APC carrying antigen. They are divided into effector T cells and memory T cells. So effector T cells circulate into the blood and reaches to the causes inflammation, killing of infected bacteria or cells, macrophages activation, killing of ingested microbes. While memory cell has a function in adaptive immunity. We will discuss it later. Since lymph node is a secondary organ, here the differentiation of T helper and cytotoxic cells occur. So therefore, we have discussed that differentiation of these cells occur in secondary lymphoid organs, while the production of these cells occur in bone marrow, which are primary. So lymphoid tissues are the tissues of the body that are divided into two, primary and secondary. Primary lymphoid organs are bone marrow and thymus. In primary lymphoid organs, the immune cells are formed, such as B cell, T cell. They are formed from hematopoietic stem cells. While in secondary lymphoid organs, naive cells, such as those cells that are not exposed to antigen, they are activated and differentiated into effector cells. Such organs are lymph nodes, spleen, cutaneous mucosal lymphoid tissues, and tonsils. So let's remove the curtain from the cytokines. What are cytokines? These are termed as messenger molecules of immune system. These are secreted proteins that mediate immune and inflammatory reactions. Cytokines involved in innate immunity and inflammation are interleukin 1, tissue necrotic factor, and a group of chemoattractants cytokines called chemokines: interleukin 12, gamma interferon, interleukin 6, interleukin 23, and several other cytokines also participate in early innate immune response. These type of messenger molecule, has an important function in the inflammation and innate immunity. Such as interleukin 1 and tumor necrotic factor causes the activation of T helper cells that causes immune response in the inflammation. Cytokines that regulate lymphocyte responses, such as effector function in adaptive immunity. These include cytokines that are involved in proliferation and differentiation of lymphocytes. These include interleukin 2, interleukin 4, and the activation of various effector cells such as gamma interferon, which activates macrophages, interleukin 5, which activates eosinophils. So this marks the end of the lecture. If you like this video, share it with your friends and subscribe the channel. We will see you in next video. Thank you for watching.